Good afternoon, everyone. My name is Mohammad Jubair Panto. I'm doing my master's in the University of Tennessee at Chattanooga, and I'm going to present my course project for spring 2022 for course long CPSC 54240 in front of you. So the topic of my today's presentation is the phylogenetic tree, the use of agglomerative clustering to understand the evolution of the Omicron sublineages. So I'm just moving forward. At first, I want to talk a bit about the overview of the whole research, what we want to accomplish, and other things. So, all we know that evolution in living organisms basically play a vital role because when organisms evolve over time, they try to change their genetic code through mutation so that they can adapt in better with the changing environment. If you consider the prehistoric era when all the organisms evolved from a single cell, those organisms were basically facing a very harsh environment, and to keep up with the harsh environment, they basically kept changing their genetic code and behavior so that they can keep pace with the changing environment and they do not get extinct. And this basically happened for the humans also. When humans were hunter gatherers a long time ago, they developed their sense of they developed their sense, and also they developed their the visual understanding in such a way that they can keep themselves safe from the other lethal animals, so which are basically which basically help try to make them extinct from the environment. But humans were smarter, and they kept changing their behavior in such a way that they can adapt with the changing environment. So the same thing basically goes for the viruses. When a specific virus changes its when a specific virus come into existence, it basically replicates and propagates and tries to transmit through the host cell. The host cell is the environment where basically viruses try to expand their population because viruses cannot live without the host cell. So the host cell environment is very difficult for the viruses to be alive because the host cell has a defense system which is called the antibodies. So antibodies try to protect the cell from the uh, outside viruses. So what they do basically, when a specific virus come into the host cell, they detect the virus and immediately kills it. But virus is very smart. They outsmart the antibodies by repeatedly changing their changing their genetic code, and that's how new variants of the virus evolve. And when a specific virus change their genetic code in such a way that it is very difficult to detect them, so that specific variant is gaining in fitness, that's why they propagate much faster, and they cause more hospitalization and death rates. So in this research, we try to find out the phylogenetic tree of the SARS-CoV-2 variants, specifically the Omicron subvariants, so that we can understand how the specific subvariants of the Omicron basically evolved over time. And that's why we used to a specific method of tree building, horizontal tree building, to construct the trees, and those methods are called Neighbor-Join and Euphoria method. And these methods are used alongside with the differential population growth to construct the phylogenetic trees. The general overview of today's presentation is, as we already talked about the abstract, then we'll be talking about this is the overview, and there will be introduction part, research objectives, methodologies, data set that you use, code demonstration. I'll be showing you how basically the code works in practice, the results, conclusion, recommendation, acknowledgement, and references. So all we know that severe acute respiratory syndrome SARS coronavirus 2 is basically a pneumoniac disease, and it is first reported on 31st December 2019 in the Hubei province of China. The most important part of this virus is that it's a single stranded RNA virus, and it has one of the largest genome sequences among the RNA viruses. And this virus has mutated multiple times after its first emergence, and mostly in the protein coding zone. There are at least 26 known protein coding mutations in this virus. This protein coding zone is very important because with the spike protein, this virus gets attached to the human ACE2 receptor, and when the virus gets attached to the human ACE2 receptor, it basically gets into the host cell by breaching the human cell barrier. And when they can do so, they basically infect the host cell. So that is why this virus changes the spike protein, because when a specific mutation occurs in the spike protein, the antibodies take more time to detect the viruses, and that's how the virus get more stealth and gain more fitness, and that's how they populate. They gain increased population than the previous variants. So due to these reasons, these specific spike protein is important for this RNA, for this SARS-CoV-2 virus, and there are total of 628 million reported cases of this due to this due to this SARS-CoV-2 virus, and till 25 February October 2022, and there are a total of more than 6 million deaths. So our research objective is to generate the phylogenetic tree of Omicron sublineages. So it is basically a tree of life where you can see how the Omicron sublineages basically evolved over time from their base variant to the other variants. So it will help us to understand the relationship of evolution, the fitness advantage of a specific variant, and how they are distinct from the other variants, and it will help us to track down their growth rate, and also track down the ancestor and the descendant variants. So it will basically help us to understand the history of the evolution, and it will add a new window to the comparative biology of SARS-CoV variants, because if we want to compare any two specific subvariants from the tree, we can just take those and compare their genome sequences so that we can understand what a specific mutation caused the two variants become more distinct than each other. And it will also help us to track down the more significant variants which are basically increasing much distinct pattern than the others. Then the specific variant will be marked as the variant of interest because it is gaining more fitness than the others, and it will help us to monitor the variants more closer manner than the previous approaches. So the methodology that we use to generate the phylogenetic tree is basically divided in two segments. At first, we used the differential population growth rate to calculate the pairwise fitness values of each and every subvariance with respect to other. Then we use that to compare the to construct the phylogenetic tree. So differential population growth rate is basically calculated by log transforming the ratio of two exponentially growing population over time. So to understand it, we can refer it to a picture. Let us consider A is the variant A, and B is the variant B, and NA by NB is basically the population ratio, rate of change of population, and we plotted it over time to understand how their growth is, and we can clearly visualize that their growth rate is exponential. Then we log transform it because to understand the fine differences in growth, we cannot refer to this picture, because if we take 200 and 800 within this region, we cannot basically understand which portion, what kind of growth rate within this time span. So after log transform it, we can see that it's a straight line, and seeing the straight line, we can have a concrete understanding that there is a positive growth rate with respect to the other. So then we did it for all the sublineages of Omicron to get the pairwise fitness values and use this to construct the phylogenetic tree of the Omicron sublineages. So to generate the phylogenetic tree of Omicron sublineages, we use two methods. The first method that we used is called the neighbor joining method. Neighbor joining method is basically a agglomerative clustering algorithm which is used to construct the phylogenetic tree of the species. And this the main focus point of this method is that it does not consider the constant rate of evolution of the species. That does mean that during the over the time species can have their variable growth rate. That means in the tree you will see different type of branch lengths in a single cluster. That is because when rapidly changing mutating viruses change, they evolve over time in different pattern. Their growth rate can be different. So that's why neighbor joining algorithm considers that they can be there can have different type of growth rate over time, and the branch lengths also can vary regarding that. So to understand the neighbor joining algorithm from the picture, we can refer to this picture. In the picture, you will see that there are 5 trees, and in the first figure, you will see there is a star-shaped trees, and there is a single node. This is basically how the neighbor joining algorithm works. At first, it starts with a completely unresolved tree like this. So you will have a node and all the taxas are here. These are called the taxas and this is the node. So they compute the distance from this specific node to all the taxas and see which two taxas have the smaller distance, and they find that F and G have the smaller distance. Then they are basically joined to each other. Then from U to other branch lengths are calculated, and to see that which specific branch taxa has the smaller distance than from this specific node. And after calculating that, they found that E has the smaller distance, so E is also joined with this. That's how the algorithm goes on and on to find the distance from this specific node to other taxas, to find out the smaller distance and add them to a new node. And after the full completion of the algorithm, the tree basically looks like this, where you can see that from each node there are variable lengths, which basically means that they have inconsistent growth rate, the inconsistent growth rate over time. That means they does not consider the constant rate of evolution. The another method that I used for this research is called unweighted pair group method with arithmetic mean. In short, it is called UPGMA. UPGMA has a very distinct way of creating the tree than the neighbor joining method. In the neighbor join, does not consider the constant rate of evolution, but in UPGMA method, they have constant rate of evolution. That means that the branch lengths will be different, will be same. Under the same node, the branch lengths will be same. That means if you refer to this picture, there are 5 taxas. So at first they calculate the distance from each other, and they found that A and B are the closest. So after found the total distance between them, it divided it by 2 because we want to consider that the branchlets are same. After assigning bracelet to this node, what it does basically, from this node it calculates all the distance from E, C and D, and found that E has the smallest distance among them, and for the total distance 22 is divided by 2 to 2, and it is 11. So the distance from this node to E would be 11, and from the distance from V to this node will be 11, but it has already 8.5 in it, so it adds another 2.5 to make it 11.11. So that's how the total algorithm works. Then it goes on and finds the distance from this node to this node and adds the closest one, and as the closest one, and divides the distance by 2, and that's how the whole tree is constructed using the UPGMA method. The dataset that I used for this research is provided by GSAID, which stands for Global Initiative on Sharing All Influenza Data. The dataset is named Variant Surveillance Dataset. That means they basically they records all the variant genome sequence submission of coronavirus in this dataset according to the location and time. And they started the genome sequencing submission for coronavirus in January 2020, and any genomes submitted to the GSite are attributed to one of these 12 clades, and that is how the GSite basically names the submitted clades. It is based on the phylogenetic distances. There are some other ways of naming the variants, like WHO named it in a way, and and Pangolinius named it in another way. There are total 13 million genome sequence submission to GSID as of October 20, 2022. As we are talking about the variant of concern, we can get a better depth understanding of the variant of concern. So mutations basically change the genetic code of the organisms, and for SARS-CoV, mutation is very obvious. And when a mutation occurs, it doesn't really change the behavior of the viruses all the time. When a specific mutation changes the virus in such a way that it increases in fatality and transmissibility, that specific variant is considered as variant of cancer, and WHO labels it because that specific variant has the higher tendency to make more hospitalization and death rates according to that situation. There are so far 5 variants of concern by WHO. They are alpha, beta, gamma, delta, and omicron. In our research, we are considering the omicron's 5 sublineages because we want to construct the evolution tree of these five lineages omicron. The five sublineages of omicron are BA1, BA2, BA3, BA4, and BA5. So, and now I will be taking you to a bit from the presentation slide to give you a visual understanding about how the code works. So, to understand the code, as I said earlier, the total process works in 2 ways. At first, we calculate the pairwise distance values from the differential population growth method. So, this is the data set. In the data set, we are considering just the variant, location, and collection rate column for our research purpose, because variant column has the Omicron sublineage enlisted in it. So we preprocessed the data to find the weekly sum of submission of each and every sublineage Omicron. So you can see that in this week there are this amount of BA1 in the was submitted to the dataset. So after getting the weekly sum of submission counts, what we did basically used the pairwise distance function that was created to find the pairwise distance matrix. So this is basically the visual output of the pairwise distance matrix. So after creating the pairwise distance matrix, we exported it into a CSV file, and after exporting it in the another notebook, what we did basically is loaded the we loaded the pairwise distance values here in the CSV format, and the distance matrix looks something like this. So this matrix basically tells the fitness values of BA1 with respect to other sub variants of the Omicron. So BA1 and BA5 are this amount of fit, BA1 and BA4 are this amount of fit, and BA2, BA1 is BA3 are this amount of fitness values of BA1 with respect to BA3 are this. So then we converted the distance value matrix into a lower triangular matrix, because neighbor joining algorithm considers it's a lower triangular matrix, because if we see in the matrix, if you just consider all the values over the main diagonal, you'll see that they're all same. There is a symmetric matrix, so the upper values are same as the lower triangular matrix. So for the calculation purpose, we converted it to lower triangular matrix. And after converting a lower triangular matrix, we use the BioPython's distance matrix and distance tree constructor object to create the neighbor join tree. And this is the final output of the tree. and we'll be talking a bit more about what the tree means in the later portion of our presentation. So I'm just taking you to the main presentation again. So this is the result of the pairwise distance matrix of Omicron sublineages. For our purpose, we created the pairwise distance matrix for North America and Europe because we want to consider the fitness evolution of the sublineages in two continents to understand the geographic variability of the evolution in different geographic contexts. And this is as I told earlier, does these values, what does these values means? This column basically says the fitness values of BR1 with respect to the other variants, and this column says the fitness values of BR2 with respect to other variants. So this phylogenetic tree of sublineages is done for North America using the Nybergen algorithm. And Nybergen algorithm basically considers inconsistent rate of evolution. That means the branch lengths will be different because they have different rate of growth throughout the time. So if you see that the branches are variable, and there are some clusters. What these clusters mean that any if you take any two taxa under a same class, same group or cluster, you'll see that they are more closer. Like BA1 and BA3 are more closer to each other than BA1, BA4 and BA5. And if you consider the total length from BA5 and BA3, this whole length, if you calculate it, it will say that the fitness advantage of BA5 with respect to the BA3, and the more the distance, the distinct the growth rate. So BA5 and BA4 are more distinct and more distant from the other taxons in the tree. That means that BA4 and BA5 had a very distinct growth pattern. And that is how the tree is read. And inner one and inner two means that this is the point where BA5 and BA4 have basically split it from the common ancestor. So this is basically the root point when they divide into two. That branch length of BA4 is not that visible here because the value is so small that it is very tough to plot. But the BA4's value is positive. That means BA5's fitness is higher than BA4. And this is the phylogenetic tree, same phylogenetic tree for Omicron sublineages, which is done for Europe, and it shows that the fitness evolution or evolution of the sublineages in the European regions. And if you see that here the BF4 length is still visible. Though the length is small, but it's still visible. That means that BF5 has a positive growth rate with respect to BF5, but BF5's distance is much higher than BF4. That means that BF5 is increasing much faster than BF4. And the tree is kind of similar like the American phylogenetic tree. That means that in both geographic contexts they have showed the similar kind of evolution. Then we use the other method, UPGMA, unweighted pair group method, arithmetic mean, to construct the phylogenetic tree of the sublineages for using the UCAS data. But the tree is pretty different from the Nybergen method because Nybergen considers the constant rate of evolution, does not consider the constant rate of evolution, but it considers the constant rate of evolution. That's why their branchlets are basically same, and it basically and from here we can deduce a very good understanding that for rapidly mutating viruses and rapidly changing viruses, phylogenetically using Nybergen method produces more accurate results than this kind of UPGMA tree because this kind of tree, this tree basically says that their growth was pretty similar with over time, but obviously BA5's strength is higher than BA4. So taking considering similar growth fitness advantage for BA5 would be an inaccurate estimation of the fitness and evolution tree. So the conclusion that we can come from this presentation is that we found that in different geographic contexts, we use Neighbor-Join method to construct the phylogenetic tree. We see that the similar fitness evolution is observed. That means in America and Europe, similar kind of fitness evolution is observed in both geographic contexts. And both figures BA5, in both figures BA5 and BA4s are closer to each other. That means that BA5 and BA4 have a very distinct type of growth rate than the other sublineages. That means that BA5 and BA4 have the higher possibility that they can lead the situation, and it has been validated by the fact that when for the time span of our research, we saw that there was a sharp rise in the cases, and which was basically the due to the BA5's BA5 subvariants. And also we can see from the tree that BA5 has a higher branch length than BA4. That means that a specific uprisal in the cases was caused by the BA5. And another deduction that we concluded from our research is that Neighbor-Join method construct more accurately than the UPGMA method. So Neighbor-Join method constructs more accurately because it does not consider the constant rate of evolution. So for rapidly changing situation viruses like SARS-CoV-2, Neighbor-Join method is more accurate because we can basically see the variance in fitness from the tree. But for the UPGMA method, we can see that the branchlets are the same, which is not that efficient in understanding the evolution of this kind of rapidly mutating viruses. One of the recommendation that we can offer is that for our research we consider the data from November 11 to June 2022. But if we can consider more data set, then we can basically see more in depth that how the fitness values of BA5 and BA4 are evolving over time, and we can also get into even more understanding that what can be the future situation regarding these two specific subvariants. And we can use a stepwise plot of fitness alongside this phylogenetic tree to understand that over the time how the fitness values are fluctuating, how the fitness values of subvariants are fluctuating. So this could be a very good visual understanding about the situation of the evolution. For this research, I am highly grateful to Dr. Chin for his continuous support on monitoring the research and commenting on the pros and cons of the methodologies and the data preparation. And I'm also grateful to SIM Center at the University of Tennessee Chattanooga for using their high computational support to carry out the research. And this research is funded by National Science Foundation, NSF. So I'm thankful to NSF for funding this research and help us to carry on the research project. Here are some important references that I used for gaining domain knowledge about the phylogenetic tree, the subvariants of the Omicron and other things. So it would be very efficient to go through these articles so that it will be even more easier to understand the total concept. So thank you everyone for witnessing my presentation for my course project. If you have any question regarding the anything about the research, you can just mail me. I will add my email address in the attachment so that we can talk in more about this. Thank you.